Good afternoon. This is Dr. Dan Guerra coming to you from Authentic Biochemistry Studios in the inland Pacific Northwest of the United States. Today is the 7th day of April 2021. And so I'm here today to give you this video presentation as the prolegomena of what I'm going to do for dissecting via a dialectical event ontological perspective of the series of audio and video lectures that I've been doing way back since the fall on aging, the immune system, and the role of epigenetics in the process of generating morbidity and then mortality in humans. So that was a pretty good introduction, I think. So let's just go ahead and start the slideshow. I think we should definitely do that. All right. So there's a picture of a tree I dropped, probably last summer, maybe 2 summers ago, out in the woods, Nez Perce National Forest. And today I'm going to talk to you about lipoproteins and acyl lipids. Acyl lipids are essentially fatty acids, usually esterified to glycerol or sphingosine. And the subtitle is Primacy in Human Biochemistry and the Pathology of Aging. All right, so let's just get into this. Now, I like this slide. It's one of my traditional ones from when I taught biochemistry every week in various graduate schools. And I want you just to take a quick look at this. There are 2 different aspects of this slide. One is lipid and carbohydrate metabolism, particularly mostly lipid metabolism in the fed state. And the second one is in the fasted state, which we should usually almost always be in because we're heterotrophic organisms, but we're also prone to be lipogenic. And because of that, we're oligonous, which means we put on depot fat in the form of visceral fat. And that means that our body can store lipid at very high concentrations because there's no water necessary to store lipid. In fact, you would need to have a lot of water if you store glycogen, for example, as your major source of carbon for bioenergetics and for biosynthesis. Storing lipid gives you that without the water, so it keeps your weight way down, you see. But because of the fact that lipid is stored in specific reservoirs called depot fat, it means that it has to be mobilized and it has to move through the circulatory system before it gets completely metabolized from the adipose tissue to muscle and obviously to the liver, where it can be metabolized in such a way that it can produce precursors to all the other macromolecules. This is what happens in the fasting state, unless you have a lot of stored carbohydrate, which you tend to get rid of pretty quickly once you start fasting. Usually by 2 to 5 days, most of the carbohydrate's gone and you're already well onto gluconeogenesis. And you're utilizing the beta oxidation of fatty acids to generate that glucose, not the carbon from the fatty acid, but the reducing equivalents of NADH and FADH2 that upon reoxidation of those 2 dinucleotides, you will generate a proton pumping ATPase generating ATP level, which will then drive gluconeogenesis, and that carbon coming from non-carbohydrate and non-lipid precursor stores. And that would include things like lactic acid and amino acids, right? Okay, so the fed state shows triacylglycerol coming from the small intestine. This is a dietary triacylglycerol moving through the lipoprotein called chylomicron, which we talked about recently in a lecture. And we also can see that in the liver, glucose and any other potential fuel will eventually, upon high concentration of those carbon sources, be used to synthesize triacylglycerol. That triacylglycerol will end up going on to very low density lipoproteins, a VLDL fraction, from the hepatocyte. And that will also get into the bloodstream. So it's 2 different sources. And then ultimately, you're going to send lipids, again via lipoproteins, to the muscle and to the adipose. Adipose for storage, and to the muscle for beta oxidation. Yes, the muscle does have intramuscular lipid droplets full of triacylglycerol with a unit membrane around them. Perilipin is usually the protein is associated with that membrane in the myocyte. And that is a good source of carbon for metabolism, for the synthesis of ATP, so that you get muscle contraction. The problem is that once oxygen levels deplete, you can't carry out complete beta oxidation of fatty acids because you need molecular oxygen. And that's when you start things like the Cori cycle and the alanine cycle, et cetera. Now, in the fasting state, things are different. I told you this is a more healthy environment for humans because we're used to having a meal and then maybe going several days without another meal. This is where your body evolved in terms of Neolithic period, where humans were going through most of their levels of metabolic speciation, probably from 1 million years ago all the way to 200,000 years ago, when Homo sapiens became dominant for the primary source of our ancestry. And then from that 200,000, 250,000 years till today, the metabolism hasn't changed that significantly. That's because it changes very slowly once it's adaptive and it works well. The problem with humans in the 21st century now is that we have readily available carbon sources because we harnessed agriculture 10,000 years ago. And because of that, we can put a lot of food by. And because of that, food is readily accessible. So we overeat. And because we overeat, we're not normally in that starve feed cycle, which is how our metabolism actually evolved during the early stages of our speciation. So what happens in the fasting state is from the bloodstream, you get ketone bodies. Like ketone bodies, remember, come from the beta oxidation of fatty acids. And they can come from various sources, right? So primarily adipose tissue, because you're fasting, so you're not getting anything from the diet. You're breaking down triacylglycerol with the help of hormone-sensitive lipases, for example, and other lipases, making fatty acids and glycerol. Glycerol can be converted to glucose, but the fatty acids, as I said, cannot. So they can be loaded onto albumin, serum albumin, and that's how they move in the bloodstream. And they can be metabolized then in the brain, in the bloodstream, and then in various tissues peripherally. And ultimately, ketone bodies are generated, for example, in the liver. And these ketone bodies, acetoacetate beta hydroxybutyrate, then can be sent to the brain because they can cross the blood-brain barrier. Free fatty acids cannot do that because they are membrane disruptive and they can cause lipotoxicity, something I talked about recently in one of my lectures, as I recollect. Okay, I think it covers everything. So when the fasted state, fatty acids are then basically being mobilized and utilized for carbon source, right? That's what we're talking about. Okay. Now, this is a much more pronounced slide to show you what's going on. Dietary nutrients, of course, enter through the portal vein. Glucose can be converted to glycogen or enter glycolysis. So here's dietary nutrients moving in. There's glucose going directly to the liver. It can be converted, of course, because of hexokinase or glucokinase, because this is the liver, to glucose-6-phosphate, and you can synthesize glycogen, the homopolymer of glycogen, alpha-1,4, alpha-1,6 linkages of that homopolymer. It is the mammalian version of starch, right? But also, of course, you can run glycolysis in the liver. Triose-phosphates, of course, can be used to generate glycerol, glycerol-3-phosphate. Glycerol-3-phosphate can be used to start building the triacylglycerol pool. But most of that carbon is going to move through glycolysis, through the pyruvate dehydrogenase reaction to make acetyl-CoA. Amino acids can give us oxaloacetic acid via transamination. And then, of course, we're removing ammonia from these amino acids. This can be from transamination to other amino acids, or it can be utilization of the urea cycle. Ultimately, though, many amino acids, but certainly alanine, can be converted to pyruvate. And then, of course, aspartic acid to oxaloacetic acid. Pyruvate can also be converted to oxaloacetic acid in the mitochondrion via the pyruvate carboxylase reaction. So pyruvate dehydrogenase, which is the decarboxylase, pyruvate carboxylase, can make acetyl-CoA and OAA, and they can run through the TCA cycle. But, in this particular version of the feeding cycle, we make a lot of fatty acid from acetyl-CoA. This would be the fatty acid synthase pathway. The FAS, of course, is itself a polymeric protein that is basically in the cytoplasm in the mammalian cell. So it's not really in the hepatocyte because, well, I mean, it's in the hepatocyte, but it's in the cytoplasm of the hepatocyte. It's not in the mitochondria. Anyways, you make fatty acids. The fatty acids esterified to glycerol. That's how you make triacylglycerol. That's the mobilized lipid in the lipoprotein fraction. You can generate very low density lipoprotein, high density lipoprotein. So where am I at on this table here? Number 5, triacylglycerol. Cholesterol can also be synthesized from acetyl-CoA, of course. So you make both cholesterol de novo in the hepatocyte, and you also make fatty acid de novo in the hepatocyte. All that can be loaded on the cholesterol then forming an ester with fatty acids, so you make CEs or cholesterol esters. You make phospholipids, of course, glycerol phospholipids, even sphingolipids, of course, with the sphingosine base. All that gets loaded onto apolipoproteins and either into VLDL or also some HDL, of course, made in the liver, and that goes back into systemic circulation. So let's run through this here. Chylomicron remnants we talked about a lot in the last few lectures. So remember that when you have dietary nutrients and you're taking in fatty acid is broken down, the triacylglycerol is broken down and then reformed from monoacylglycerol, and fatty acids are reattached. The reason that is you need a detergent generating event process, so that you can correctly organize the chylomicrons, which are going to be your first lipoprotein fraction. And remember that in order to do that, those chylomicrons have to coalesce with the apolipoproteins, which we'll get into more detail in a moment. But you can make all these different lipids here from the chylomicron remnants. It's going to deliver lipid from nutrition from the small intestine, right? It's going to give you free fatty acid, diacylglycerol, monoacylglycerol, phospholipid. It's going to bring you dietary cholesterol as well. So there'll be some biliary excretion of cholesterol end products. But basically, we're looking at the lipoprotein here. We're focusing on that. And you're running all that lipid back out of the liver. Now, if you don't, and you store a lot of lipid in the liver, then you can get a fatty liver, which can cause all kinds of problems down the road, particularly as you age. The non-alcoholic fatty liver disease, and the FLD, can lead to non-alcoholic steatohepatitis, which can lead then all the way to hepatocellular carcinoma and cirrhosis of the liver, either one of those two, either cancer or cirrhosis. And sometimes cirrhosis can lead to cancer. And that, of course, damages enough liver. If you lose your liver, if you don't get a replacement, you're going to be dead. But if you get a fatty liver, it can cause a lot of oxidation of those lipids as storage products, storage droplets inside the liver, and that can wreak havoc on the normal processing and metabolism that is supposed to occur in the hepatocyte. So that's one of the things that happens as you age. Right? All right, so let's see if I covered all this. Amino acids and the amino acid pool, transamination, as I said, short-chain fatty acids bound to albumin, that's serum albumin, and then you get reincorporated into chylomicron remnants, for example. Chylomicron remnants attached to ApoA, that's the lipoprotein that's necessary to coalesce those lipids once you move through the monoacylglycerol pathway. And they are endocytosed and taken up by the lysosome. And that's where you get all the free fatty acid, monoacyl, diacylglycerol, et cetera. I just mentioned two cholesterol esters. And the rest of this, I think, has already been described. VLDL delivers the meals, lipids to non-hepatic tissue. There's got to be a lot of interaction between VLDL, HDL, and the other lipoprotein fractions. That's going to be happening in the periphery, both in circulation and in different cell beds, right, tissue beds. All right. So that, I gave you a couple cartoons at the beginning because I'm going to give you now some text. So aging is marked by a progressive decline in the function of multiple cellular lineages and tissues that come from them, of course. In organisms with renewable tissues, aging is also marked by an increase in hyperplasia, the most serious of which can be what? Cancers, right? So why does aging occur? Right now, we can ask that question. Remember, that it's the Prolegomena for the full-on lectures I'm going to give you. That it's going to present all the previous work we've done for the last several months and coalesce them like a lipoprotein into a couple, 3 video lectures. So it's the Prolegomena lecture. Evolutionary theory holds, aging, okay, we're talking about aging now, that aging is a consequence of the declining force of natural selection with age. Okay. There are extrinsic hazards that always occur in any living system. There could be accidents, predation, infection, starvation, and all of that will limit the lifespan of any species, really, except if you're talking about microorganisms, and there you're not really talking about individuals, right? You're talking about colonies of cells. But what happens is that will deplete natural populations of the older individuals, which will allow younger individuals, in our case we're talking about human aging, to then, because they have more muscle strength and their minds are sharper, to be able to carry on with generating a humankind, right? So there are generally few old survivors on which natural selection can act to eliminate alleles or genes that have late-acting and, of course, very deleterious effects. So once a person reached to be 70 years old, male or female, most of the processes that are controlling the constant source of reactive oxygen, which can cause either apoptosis and therefore degeneration of cellular systems and senescence, you can run the risk of getting mutations, of course, in DNA, RNA, lipids, in the lipids that are generated from enzymatic activity, and all of those mutations can wrest the system from its homeostasis into a rapid, highly morbidity prone aging process, which can lead ultimately to mortality, death, right? And there are really few older survivors that are going to be reaching that climax of the life. But because of the fact that biomedicine has made such advances in the last 100 years, the lifespan of humans has increased significantly. Part of that also, of course, is nutrition, right? But the problem with it is that the body may be extended, particularly with surgeries and with pharmaceuticals, but the mind does not owe such a significance to the future fate of an aging process. And so what you end up with is a high level of morbidity that ends up being neurodegenerative, right? And that's why we see increases in Parkinson's and Alzheimer's disease, prefrontal dementia, etc. The body can be sustained and maintained longer than the central nervous system. That's because the central nervous system is far more fragile, because the output of the central nervous system, that is the mind, is easily detected as degenerating because of memory and all the foci that are normally associated with self, right? Whereas if muscle cells are slowly deteriorating because of sarcopenia, or vascular beds start to degenerate, atherosclerosis, and degeneration of the pathways for respiration, for example, a person slowly declines in those areas. They can't run as fast, they can't lift as much, they can't do as much physical exercise. But when the brain has this dysfunction occurring, it's much more obvious to the rest of us that are not experiencing that dysfunction, especially if it's someone that has generated, right, a transcendental self over time, and that transcendental self, that mind, starts to deteriorate in front of our very eyes. And that has to do, again, with the body degenerating, the periphery of the body degenerating probably at the same rate as the central nervous system. But the readout of the central nervous system is something much more acutely recognized by the rest of the human population. And that's why we notice it. Right? Okay. So anyway, deleterious effects, especially true for genes that confer early life benefits. That means, of course, reproductive capacity, and of course, this whole thing about having a tone and a higher athleticism when you're younger. So natural selection cannot eliminate genes that promote early life survival, but incongruously also promote late life debility. So we have a name for that in the aging literature. We call that antagonistic pleiotropy. So let me repeat how that works. Natural selection probably can't eliminate genes that promote early life survival, which clearly they probably do because of selection on the hoof. That is why you're living. but also genetically. It would be more epigenetically, right? But incongruously, you can't have that also promoting late life at the same level. So late life debility or disability occurs because what promotes early life survival may work just the opposite, be actually contradictory to late life survival. That is, for example, a really active immune system. So a hyperimmune response, or perhaps a very deficient immune response, is going to increase morbidity and therefore mortality as you age, you see. And I just mentioned to you already about the central nervous system degeneration, right? And don't worry, we're going to get into that in some great detail. Now, aging phenotypes. The most prominent feature of aging is degeneration. Degeneration gives rise to well-recognized pathologies, such as sarcopenia, that's muscle wasting basically, atherosclerosis, right? We know a lot about that now because I've given multiple lectures on that over the last year. And that can lead to heart failure. That's not a heart attack, it's just heart failure. But you also can get osteoporosis, right? That has to do with mineralization, bone remineralization. So your ultrastructure becomes more porous and more prone to breakage, right? And unable to carry around the weight, which may be increasing because of the increase in depot fat, visceral fat. And that's because of overeating, because of the invention of agriculture I mentioned 10 minutes ago, right? You also get kidney failure, renal failure associated with another common morbidity in aging. Neurodegeneration, that includes prominent neurodegenerative diseases, which I've already mentioned. Alzheimer's disease, Parkinson's disease, prefrontal dementia, things like that. Degeneration in one or more tissues is an extremely common and prominent age-related phenotype. Okay. So you also get tumorigenesis in aging, right? Among multicellular organisms with renewable, that is repairable or regenerative tissues, aging entails another feature, that gain of function changes, which can allow cells to proliferate inappropriately. And that we call that hyperplasia. But if it gets completely uncontrolled because of mutation, and then it starts to break through basement membrane and become metastatic, then we call it cancer, metastatic cancer in particular. So through genomic instability, these changes allow cells to acquire phenotypes that increase their ability to proliferate, migrate, and colonize ectopic sites, to survive hostile tissue environments, and to evade the attack by the immune system. That would be the hostile tissue environment, right? So these phenotypes are emblematic of terminal cancers, right, of the aged. So aging then can lead to an accumulation of random epigenetic and indeed genetic alterations that can give you increased probability of oncogenic events. Aging can also cause a decreased progenitor cell fitness because you're not synthesizing as many, for example, neurons, right, or muscle cells, including cardiovascular heart cells, right? You also, from aging, get altered systemic and microenvironments, right? This has to do with reactive oxygen. You also get altered immune responses, as I mentioned, because of the proliferation of T memory cells and the decrease of T naive lineages, right? Coming from the lymphatic system, coming from lymphoid organs, right? Also because of the slowing down and degeneration of the thymus, right? Where T cells can be recruited, amplified, and programmed via education not to attack self. All of that decreases over time. So that altered immune system gives you either a hyperimmune response when it's not supposed to occur, thus causing autoimmune disease. And I would argue many of the diseases of the elderly really are autoimmune diseases, such as neurodegeneration, tissue degeneration, even atherosclerosis to some degree, because that's certainly an immunogenic causal environment. But also, of course, you can get, besides all of that, the degeneration can also get oncogenesis. So you get a promotion, expansion of mutant clones through all of these different deteriorating pathways, and that can all lead, because of an increased probability of oncogenic hits, as you live because of more and more reactive oxygen, because we live in an oxygen-rich environment, cancer. So cancer risk in aging is shown here in this graph. This is from the National SEI. Number of cancer cases per 100,000 people you see has been generally increases as you age. For example, breast cancer and colon cancer, very common forms of cancer in humans. Now there's a stress response termed cellular senescence, and that links multiple pathologies of aging, both degenerative and hyperplastic, as I just mentioned. Cellular senescence is unlikely to explain all aging phenotypes, however. Nonetheless, a surprisingly large number of aging pathologies have been linked directly or indirectly to this senescence response or phenotype. So you have normal cells or just plain young cells, and they can undergo genetic alterations, mutations or deletions in the genome that affect gene expression. You can also get an increase in copy number of the genes, right? All that can then lead to aging and cancer phenotypes being initiated. There's supposed to be a cancer cell here. And then you get epigenetic alterations. This basically has to do with signature changes in gene expression because of methylation patterns, acetylation patterns of chromatin, such as the methylation or acetylation of specific nucleotides that are in promoter region or enhancer regions for chromatin remodeling associated promoter inducing expression of genes, right? Or it can also lead to simply just malformations in the way genes are spliced, and because of incorrect splicing or splicing variants, that can also lead to changes. That's part of the epigenetic alteration. There's also acetylation, methylation, sumoylation, ubiquitinylation, various other covalent modifications of histones, which can also corrupt chromatin remodeling. Chromatin remodeling, of course, associated with gene expression. So that's all shown over here. Changes in the chromatin structures that affect gene expression and function are all basically known as epigenetic. It just means that you're not getting an alteration of particular DNA code, right? So the triplet codons aren't necessarily being corrupted by mutation, although that can happen too. That's over here. But this has to do with alterations of the chromatin itself. Also an alteration of microRNAs. MicroRNAs are small RNAs, small single-stranded RNAs that regulate transcription and translational processes, therefore gene expression. So you get aging and cancer phenotypes initiated from that mode too, from epigenetics, from genetics. You get further genetic alterations or epigenetic alterations. You see now there's more genetic alterations here because these are mutations on an epigenetic vestibule. And here you have a genetic vestibule and you get epigenetic alterations, again, for the same reason, alterations in histone, covalent modification of histones or covalent modification of, for example, cytosine residues and CpG islands, promoter regions of DNA, thus altering gene expression. Methylation is one of the key features there. So then you have to think about the levels of acid-N-acetyl methionine, which comes from folic acid metabolism, which is also, of course, linked to aging. And then you get progression of aging and cancer phenotypes from this. This is a normal state of affairs, right? So then you get what's called the senescence response. Causes can be persistent telomere shortening, genomic damage, right? So DNA repair can't keep up with the amount of genomic damage. You get oncogenes that are expressed. You get strong mitogenic signals. So again, you're dealing with increases in mitosis, increases in cell division, which are supposed to occur. The epigenetic permutations and perturbations I've just talked about, methylation, acetylation patterns, etc. Tumor suppressor gene activation. Then if those tumor suppressor genes have been mutated, they can actually bypass the normal cell cycle and then enhance, because of senescence itself, a proliferation of cells. Consequences then, you get some tumor suppression. You also get some tissue repair, age-related tumor progression, and age-related degeneration, okay? That's basically what this says down here. Now, genomic and epigenomic stress can also lead directly to different types of DNA repair, DNA damage repair mechanisms. A persistent DNA damage repair system can lead to P53. P53 will cause growth arrest. This is part of the chromatin a halting of cellular division by p53, right? So this is going to stop cell cycle. But you also get, because you get persistent DNA damage repair, you also get this senescence-associated secretory phenotype, which p53 can block, but SASP, as we talked a lot about, can also sometimes lead to growth arrest, but also, unfortunately, can signal to other cells, generating a pseudosenescent phenotype, which actually allows them to exist and last longer, but generating more and more mutations, which ultimately can lead either down the pathway of neurodegeneration, for example, central nervous system, or down the pathway of oncogenesis. Right? So any kind of other stresses besides the genomic and epigenomic, this can be caused by any number of genotoxic agents or epigenetic agents that are otherwise related to things like reactive oxygen or perturbations in diet, or obesity being a key function there. This turns on this P16 pathway, which turns on the PRB pathway, and this will allow for the synthesis of heterochromatin. So again, this is basically a deacetylation pathway from euchromatin to heterochromatin that also causes growth arrest. Okay, so you can, when you go back over this slide, you can read all that. It talks about NF-kappa B and more of the players that are involved in this process. So I'm not going to read all that to you. You can read it at your own leisure. So cellular senescence refers to the essentially irreversible arrest of cell that occurs when cells experience potentially oncogenic stress. The permanence of the senescence growth arrest enforces the idea that the senescence response evolved, at least in part, to suppress cancer. Right? But if you have pharmaceutical and surgical and dietary intervention and you prolong life, it doesn't necessarily mean that you're also suppressing at the same time cancer. Remember that pleiotropic antagonism I told you about, right? So you have an aging system that you're trying to keep young, but at the same time, that aging system has got its own sequelae of effort, and that is generating more senescent phenotypes in cells. And when that happens in the central nervous system, that can wreak havoc throughout the entire body, right? So senescence arrest is considered irreversible because no known physiological stimuli can actually stimulate senescent cells to re-enter cell cycle. So once you get senescent cells, they're senescent, that means they're not going to divide any longer, and they're basically eventually going to die, right? And they're going to lose function while they die. However, molecular biological manipulations, for example, the sequential inactivation of tumor suppressor genes, you can do this pharmacologically, can cause senescent cells to proliferate. Now, the same thing can happen because of just the normal vagaries of disruption of metabolism, such as caused by dyslipidemia, linked directly to obesity, right? Which is a very common, terrible pandemic in humans. Between 60 to 80% of humans now worldwide are either prodromal for obesity or are full-blown obese. This can lead to a whole constellation of diseases that we're seeing in late in life, but also increasingly in early life. But that morbidity lingers longer because people stay alive longer because of things like surgeries and pharmaceutical interventions and dietary interventions. You see. So, molecular biological manipulations, for example, sequential inactivation of tumor suppressor genes, as I said, can cause senescent cells to proliferate. There may be as yet unrecognized physiological circumstances or consequences under which a senescence growth arrest is reversible. We don't really know if that's the case. We see this in some model organisms, but it just hasn't really been well documented in human beings. And that's what we're talking about here, human metabolism, human aging. Now, regardless, the senescence arrest is stringent. It's established and maintained by two major tumor suppressor pathways. The p53-p21 pathway, remember these are not going to be checkpoint level on the cell cycle, not checkpoint on the immune system, right? That's a totally different thing. That's the PD, PDL, and the CTL4A. Here we're talking checkpoint, classical, canonical checkpoint, on cell cycle, right? That's p53, p21 pathways. But there's also the p16 INK4A, PRB pathway that I talked a great deal about last fall. And that's why I'm bringing it up again now. And it's now recognized as a formidable barrier to malignant tumorigenesis, right? So there are major tumor suppressor pathways, and they do tend to tamp down the malignant process. Consistent with that view, cells undergo senescence in response to a host of potentially oncogenic stimuli. So you can do this in cell culture. You can induce an oncogenic event. You can cause a genotoxic or an epigenotoxic event, and then you can determine whether or not those cells go into senescence. And if they do, they can prevent the oncogenic lineage to develop, right? Because you go senescent rather than oncogenic. This is believed to be part of the evolutionary process. Now, in addition to that, to arrested growth, that basically what senescence is going to give you is not going to divide or expand. Senescent cells show widespread changes in chromatin organization and gene expression themselves. Of course, right? You're getting a senescent phenotype, which isn't the same as the youthful phenotype, okay? In terms of gene expression, in terms of lipid profiles, things like that, right? Now, these changes include the secretion of a huge amount of pro-inflammatory cytokines, chemokines, and these don't just come from immune cells, these also come from epithelial cells. But you get growth factors, which can stimulate certain immune responses, proteases, which also are involved in immune responses, right? This is the whole intracellular pathway and extracellular pathway that we talk quite a bit about, like B cells utilize protease activity when they're generating the pathways for removing foreign agents or foreign cells from circulation and from tissue beds, right? So that's a whole process. So you got proteases at work, growth factors at work. That's what we're talking about. And we've discussed these proteases. We also talked about protease inhibitors like serpins, right? Serine protease inhibitors, and then serine protease inhibitor antagonists being used for cancers. We've been well into this discussion, I'm sure, because you remember, because I remember. Anyways, all of this changes in secretion of all this potential hyperimmune response is called the senescence-associated secretory phenotype, or SASP. I talked about it a great deal. Okay. So SASP then, angiogenesis, cell proliferation, resistance to chemotherapy because it's senescent, epithelial to mesenchymal transition, we talked about this as well. It's called the EMT pathway. Stem cell renewal and differentiation that could potentially become oncogenic down the road. Inflammation, hyperinflammation, in fact. And then, of course, tissue repair, keep things going, right? So there's myriad of activities, glasses on here because it's difficult to read down there, of senescence-associated secretory phenotype. It's secreting these pro-inflammatory cytokines, for example, growth factors, etc. The many factors that compose the SASP have numerous biological activities, all highly dependent upon physiological context. That is it an aging context, or is it a prodromal phase of some disease? The activities include stimulation of angiogenesis, we just said, stimulation and inhibition of cell proliferation, on or off, creation of the chemo-resistant phenotype using during cancer chemotherapy, right? Stimulation of an epithelial mesenchymal transition, all these things, right? Okay. Which we've just talked about. Now, the causes of cellular senescence. Now, there are more than these, but this is a good list. Cellular senescence was first formally described approximately 5 decades ago when Hayflick and his colleagues showed that fibroblasts did not proliferate indefinitely in culture. These cells were said to have a finite replicative lifespan, that is division, and later to undergo replicative or cellular senescence, and sometimes that's termed replicative or cellular aging. Right? The number of divisions that cells complete upon reaching the end of the replicative lifespan have been termed the Hayflick limit. Okay? The link between the Hayflick limit and aging was for many years conjectural and tenuous, largely on the basis that replicatively senescent cells appear to be quick to degenerate, although they remained viable and metabolically active. It's just that if you looked at what kind of metabolism is going on, there's a shift in carbon utilization between carbohydrate and lipid. That's one of the things to keep in mind. And 50 years ago, it was evident that most cancer cells do not have a finite replicative lifespan, so they blow right past the Hayflick limit. Hence, the idea that the senescence response is tumor suppressive, right, in conjunction with our understanding of what suppressive and senescent phenotype looks like in terms of its regulation, although still speculative 50 years ago, is becoming more firmly grounded now in the 21st century. Lipids control, yes, lipids control cell fate by regulating membrane permeability, polarity, and indeed signaling. Key features of lipid metabolism in mammals, of course, bioenergetics, beta oxidation of fatty acids, and also the utilization, as I said, of the oxidation of those fatty acids to generate the reduction of NAD and FAD to NADH and FADH2, and their reoxidation electron transport chain to make ATP for gluconeogenesis. And then that glucose can be used for bioenergetics. So trade-off among carbohydrates, amino acids, and of course fatty acids. And that shifts, all of that will shift the immune response because those changes in metabolism are occurring inside lymphocytes and both T cells and B cells, but also in the innate immune cells, in macrophages, and in other antigen-presenting cells, such as dendritic cells, and also in neutrophils and mast cells, and all these different innate or acquired immune cells are altering their carbon source, their bioenergetics, either with fatty acid, amino acid, or carbohydrate, and all of this then coalesces into or funnels into the telescopic means to either be a senescent phenotype or to stall that or to replicate that because of long distance signaling. That has to do with the secretion phenotype. Right? Okay. So key features of lipid metabolism in mammals is bioenergetics, trade-off, we just said all that. Oh, I didn't mention this down here. Lipids shift the immune responses, and from a surveillance pathway, which is considered healthy, to agentic effector inflammatory response. Okay? This is not a good thing. This can lead to hyperinflammation. Aging results in dyslipidemia. Now, if you're already obese, that dyslipidemia comes on much earlier. Therefore, it can be a prodromal aging phenotype. That is obesity. Membrane deterioration, of course, because of reactive oxygen, among other things, and free fatty acids moving through the membrane will cause degeneration. That, in fact, is the component of lipotoxicity. And that links lipid metabolism to senescence phenotype and excessive oxidative metabolism. And all that leads to morbidity and, yes, to death. So, take a look a little bit at fatty acid metabolism. Fatty acid biosynthesis can occur in the liver, as can fatty acid oxidation. Citrate breaks down in the cytoplasm. Citrate is generated in the mitochondrion. Once it leaves there, citrate lyase will generate acetyl-CoA and oxaloacetic acid. That acetyl-CoA then can be used to form malonyl-CoA. That's the acetyl-CoA carboxylase reaction. That's the rate-limiting step for de novo fatty acid synthesis. Okay, it's activated. That ACC, the acetyl-CoA carboxylase enzyme, is activated by citrate. It causes a polymerization of the enzyme, making it far more active. But palmitate, which is an end product of de novo fatty acid synthesis, coming directly after thioesterase reaction of the FAS, that palmitate then will inhibit acetyl-CoA carboxylase. Okay, so these are allosteric modifications. You also get inhibition by cyclic AMP-dependent phosphorylation of the enzyme, because obviously with cyclic AMP, you're talking about a fasting state or a stress state. Whenever you make cyclic AMP, that means that you don't want to be synthesizing fatty acids. You want to be mobilizing reserves, right? Not synthesizing long-term storage lipid, which acetyl-CoA carboxylase ultimately is going to govern, right? And you also get activated by insulin-dependent dephosphorylation, because insulin is a well-fed state, so you do want to go through lipogenesis. You want to make lipid, send it out with the lipoproteins into the adipose tissue and build your visceral fat, build your depot fat. For the long period of time, you should be experiencing fasting or even starvation, because I told you that's where your body evolved from. Right? So anyways, if this is functioning in this correct manner, triacylglycerol amongst cholesterol esters and phospholipids, including glycerolipids and sphingolipids, all get loaded onto very low density lipoproteins, and they can make it various places. Here it's showing you being taken into the adipocyte, right? And then there's a lipoprotein lipase that's going to release some of the free fatty acid, and that's going to reform the triacylglycerol. It does that, again, for a detergent effect in the adipose, but also so that those fatty acids can be used for other modifications within the adipocyte, such as acylation of proteins, right? It's one of the reasons you're probably breaking down those triacylglycerols. And plus, you're reforming a certain molecular species of triacylglycerol that stores better. And this has to do with the level of polyunsaturated fatty acids in the 1 or 2 position of glycerol lipids. Right? The more polyunsaturated fatty acid, more auto-oxidation can occur, not good for visceral fat. So storage of triacylglycerol is better if you have either monounsaturated fatty acids or saturated fatty acids in depot fat. There's a lot of other mediators of micellar formation, and also the ultimate mobilization of the lipids when they're called upon, that will yield a more friable product for mobilizing fatty acids upon the signaling that generates the fasting response, and therefore the requirement for fatty acids to be loaded onto lipoproteins or serum albumin sent to the liver for gluconeogenesis. Okay. So there's a lot of membrane-mediated responses there, which we have talked about in the past, and of course I will again. During fatty acid oxidation, when lipids are released from the adipose, right, hormone-sensitive lipase, which of course is activated, the way you shut off acetylcarboxylase, you turn on the lipase here in adipose, cyclic A and B-dependent phosphorylation, hormone-sensitive lipase is kicked on. That's going to take fatty acids generated from triacylglycerol, mobilize them from the adipocytes into the bloodstream. They're going to make it in the liver, where you're going to then carry out beta oxidation, as I said. First, you have to esterified acyl-CoA. You have to run those fatty acids in the mitochondria. That's the carnitine transferase pathway. And then you break down fatty acids via the oxidation pathway, making acetyl-CoA to the ketone bodies, acetoacetate beta-hydroxybutyrate. Also, some citric acid is generated. And then again, that you do actually, the liver can walk and chew gum at the same time. It can synthesize fatty acids even when it's generating ketone bodies. And that's because there's metabolic zonation in any good complex organ. And the liver is a very complex and very good organ unless it becomes inundated with too much depot fat inside it, right? And also cirrhosis, and people who drink alcohol can certainly generate that degradation of the liver and get very ill and die very young, which is really sad. And also drugs like amphetamines and cocaine and even marijuana to some extent can cause problems with hepatic metabolism. And we talked about that in the past in Authentic Biochemistry. And don't worry, I will again. This is kind of just a classical lipoprotein, what they look like. Remember, lipoproteins are non-covalent associations of lipids with proteins. So if you want to talk about covalent associations, those would be proteolipids. So lipoproteins are non-covalent. Now, there is some covalent modification of proteins, but for all intents and purposes, it's a non-covalent coalescence of lipid with protein. So the core contains triacylglycerol and cholesterol esters. That's very hydrophobic. Phospholipids on the outside because they are somewhat amphibolic, so they can interact in the circulation, for example. And you have apolipoproteins, which are all embedded with in and without. And those are the key signatures for the different subclasses of the lipoprotein fractions. And then, therefore, the kinds of lipids that will associate and the density, right? And that has to do with these proteins were first isolated, or we talked about this, they were isolated via ultracentrifugation. First, they were done on starch gels, but also on ultracentrifugation. And the density of the protein was the first thing that was noticed. That's why there are things called VLDL, LDL, IDL, HDL, right? Because that's how they were discovered chromatographically or via centrifugation. That's why they're called that. Different densities, obviously more lipid, less dense, less lipid, more dense. Right? Okay. Now, this is a really important slide. This shows you basically what happens in the atherosclerosis. So it's a very busy slide, but I like it. Highly oxidized aggregated LDL is formed in the vessel For example, in a vessel that's pumping blood, right? For example, to the heart. And as a result of that action of reactive oxygen species, you get enzymes that are degradative, like sphingomyelinase, that's going to generate ceramide. Ceramide is a very potent pro-inflammatory molecular species of sphingolipid. You also get secretory phospholipase A2. That's going to release free fatty acid from the 2 position of glycerolipids. That fatty acid can then be oxidized, generating the oxy fatty acids, right, known as the eicosanoids. These fatty acids are pro-oxidative themselves, chain reaction phenomena which can generate a highly toxic oxygenic environment in the vascular bed, shown here, right? So the oxidized aggregate LDL is recognized by the macrophages, which are also embedded in this system, in this vessel, right? They're taken up, macrophages are taken up, the LDL is taken up. It says you're showing you macrophages in the system, showing you how macrophages are interacting with removing cholesterol to the HDL fraction, which will get rid of cholesterol. You have extracellular lipids within the vessel. These extracellular lipids are also going to be a source of reactive oxygen. It's going to generate necrotosis and apoptosis. This is all going to be very bad. Also ferroptosis. All of this is going to be really bad because it's going to cause the production of aggregates. Those aggregates are going to start causing atherosclerotic plaque formation, right? This will then cause an occlusion of the vessel, which can lead to heart failure, heart disease, right? That's what we're looking at here. Myeloperoxidase, lipases, reactive oxygen, all generated because of macrophage normal biosynthesis of reactive oxygen. For example, via the NADPH oxidase pathway, because macrophages, remember, are innate immune cells. So what are they doing in here? They're generating toxic substances like hydrogen peroxide, superoxide, even hydroxyl anion radical. All of that's going to cause further oxidation and enzymatic modification of LDL. So you have oxysterol LDL. So it's not the cholesterol that gets you, it's the oxy LDL. And that's a molecular phenotypic species of LDL, which does cause the atherosclerotic plaque. So you get foam cells thus forming because of this degeneration of the macrophages. These foam cells secrete another protein called apolipoprotein E, which can actually lead to some forms of Alzheimer's disease if this is happening in the vascular beds of the central nervous system. But only certain mutations of apolipoprotein are associated with that, so not all. But inside the vessel, they can wreak havoc. They can facilitate removal of excess cholesterol, which is a good thing. Now, that can happen with, of course, the HDL fraction, but it doesn't always happen, particularly when you get all this reactive oxygen being generated. Now, all this CD6, 68, CD36, this is the transport and movement of fatty acids in and out of these different cellular lineages, which are all in the vessel, right? And because of that, you get more reactive oxygen, you get more lipid toxicity, all occurring because of this enhancement of lipid moving in and out of circulation. Now, where would you get all this occurring? In the obese phenotype. The obese phenotype, you have really high levels of circulating lipoproteins with various stages of oxidation. These oxylipids, including oxysterols, can lead to this occluded vessel, leading to cardiovascular disease, which of course is a very common cause of death in humans, that or cancer, right? So you get the idea. And of course, this can be potentially oncogenic because of all the reactive oxygen causing mutations in DNA, in protein, and in RNA, thus altering gene expression, thus leading to a pro-inflammatory phenotype, which can completely corrupt the system, leading to degeneration or oncogenesis. Okay. So overall, a very, very bad state to be in. This is what happens over time in the aging human, though. It's not. It's a pathology, and there is a pathophysiology associated with it, which has been well discussed and documented in the literature, and I've talked a lot about it. But you can exacerbate this if you have an obesogenic environment. I told you between 60 and 80% of the world population is either prodromal obese or are full frank obese. And you get the idea that all of this disease is then carried on into old age. And even with pharmaceutical intervention, with surgical intervention, this leads to high levels of morbidity because of drug-drug interactions for all the drugs that people are taking because of their obesogenic state, and perhaps type 2 diabetes, metabolic syndrome also playing a major role here. And then that leading to things like central nervous system disorders such as Alzheimer's, Parkinson's disease. All of this is linked. All of this is canonical and associated in very complex ways, which we understand, but which cannot be easily diagnosed from one human to another because of epigenetic modifications, dealing with changes in gene expression, which makes every human being absolutely unique. So again, it shows you the same thing I showed you before, the regulation of the pathways. I did mention that malonyl-CoA, which is the central product of acyl carboxylase, which is the basically rate-limiting step for de novo lipogenesis in mammals, in fact, in all cells when I think about it. Malonyl-CoA acts as an inhibitor of the carnitine palmitoyltransferase pathway. It doesn't allow fatty acyl-CoA to make it into mitochondria. Right? So once malonyl-CoA builds up, that doesn't simply mean that if it's there, because it's there as a product of acyl carboxylase, but as a substrate for de novo fatty acid synthesis. Right? So it doesn't. If it builds up, it has to build up because there's an excessive amount of acyl carboxylase activity, and not an equal excessive amount of utilization of the malonyl-CoA, for example, in fatty acid synthesis. Right? And that could be because of corruption of the pathway, mutations, which can be associated with aging, but also just with a slowing down of the fatty acid synthetic machinery because it is a complex series of reactions all loaded onto one polyprotein, which is cytoplasmic, again, in humans. All right. So we already talked about this too, the homosensitive lipids, just a blow up of this. So what's the role of the liver and adipose tissue in lipid metabolism? So the metabolism of triacylglycerol during fasting. The adipocytes, you have lipolysis, homosensitive lipase can generate that. That will release fatty acids into the blood, binding up because it's a lipid. Fatty acids are not going to be in free circulation. They're going to be bound to a protein non-covalently, such as serum albumin. The adipocytes are carrying out this process, right? And what's the liver doing? It's by basically producing ketone bodies during fasting. It can even between meals make ketone bodies. It's the whole idea of ketogenic diet, which is way overblown and way misunderstood in popular culture, popular media, and even popular science. And popular science is an oxymoron. There's only research science, so they'll use the word science itself as kind of foolish because science is more like a philosophy than an endeavor. So we talk about research science. That's a totally different thing. In fact, research is the way that we gather up all the information I'm telling you, right? It has nothing to do with the downstream processing of some research phenomena that can lead to biomedical activities. Always keep that in mind, right? Biomedicine is served by research, and research is very seldom conducted by medical doctors, right? Very, very seldom. Most research is carried out by PhDs. Well, we do most of the basic research. We also do most of the education of the MDs. Need to put that in there for the obvious reasons. Now, metabolism of lipoproteins. You have the LDL. You've heard of this probably, and forget about everything you've ever heard in popular press because I'm going to explain it to you correctly and authentically because I'm a lipid biochemist and this is authentic biochemistry. So, QED. Low-dose lipoprotein transports cholesterol to tissues, so that's a good thing. There's nothing wrong with that. Cholesterol needs to be sent to tissues because cholesterol is a precursor to a whole host of steroids, right? And you make a lot of cholesterol in the hepatic tissue, so moving it in lipoprotein fraction to the serum is a very good, normal physiological process, not pathophysiological. And LDL binds with LDL receptors, so the whole idea of LDL receptors, their regulation, their turnover also can lead to some pathologies, which we saw a couple slides ago when we were talking about the atherosclerotic plaque formation. LDL receptor itself has a specific structure, certain protein fractions associated with a movement of the LDL receptor to the plasma membrane, and that movement is going to be associated with the production of lipids through the endoplasmic reticulum, through the Golgi apparatus, thus loading the LDL receptor and recycling it back and forth in the plasma membrane. And that's going to be associated with steroidogenesis and frankly, cholesterologenesis in those cells. So the whole process itself, LDL receptor turnover and cholesterol metabolism, is linked also to cholesterol uptake in certain cell lineages. Right? So mutant cells can't bind efficiently, synthesize cholesterol to meet the needs. So free cholesterol in the cell serves multiple regulatory functions. One of them, during de novo cholesterologenesis, is you make geranyl-pyrophosphate, farnesylpyrophosphate, and the geranyl and farnesylation of proteins are often what precludes the movement of receptors to the surface, because receptors are often prenylated and acylated. Without the correct precursors in the cholesterogenic pathway to load those receptors into the membrane, you get a malfunction then of the receptor-mediated endocytosis of the lipoproteins, you see. And this can be corrupted by statin use in ways that sometimes shows up phenotypically and pathophysiologically, and sometimes not, and sometimes a cult. Okay? And we've talked about that a lot in Authentic Biochemistry, and don't worry, I will put that in as a major section when we get into the dialectical event ontology of aging, the immune system, and the epigenetic profiling as we move from morbidity to mortality in humans. So I'm going to stop here because this is where we really start getting into lipoprotein metabolism. I told you this is Prolegomena, and I did kind of like trick you by saying we're going to talk about lipoproteins, but we did. At the very beginning, I talked to you about chylomicrons and chylomicron remnants and LDL, VLDL, and HDL. So I did do it actually throughout. I got into it more intensely in these last few slides. But the true canonical discussion of lipoproteins is going to come in the very next video lecture. So yes, you're going to get to see another video lecture and not just hear me on the audio for 29.8 minutes when it gets cut off, because that's the platform I use. So with video lectures, obviously, I can do more. Plus, you get to see these slides. You don't just hear me speaking real rapidly. And you also get to see me, and you see that this is basically me giving you a lecture, right? All right. So I'm going to stop here, as I said, as I threatened. Let's get this right here. All right. So let's get rid of that. All right. Now this is all my recordings for the Authentic Biochemistry lectures I've been doing on video for the last couple of years. So all we really need to do now is tell you that we'd like to see you again next time. I'm hoping I can make another video lecture this week, probably 2 more this week. Today is Wednesday, as you know, the 7th of April. I'm hoping to make another one either Thursday or Friday, and then another one over the weekend. And then we'll probably have 2 more, maybe 3 more, which will be the full-blown dialectical event ontology of this series of lectures. And then we will actually, maybe by May, be completely finished with this long arc of aging, the immune response, and the epigenetic profiling of morbidity, mortality in humans. Maybe by May. And then I've got a really, really cool new topic that's going to come up, but I'm not going to tell you what it is until we get there. Because I like to keep some things, exciting. And exciting means I keep some things hidden from view until they're ready to go. Okay. So this is Dr. Dan Guerra from Authentic Biochemistry Studios in the inland Pacific Northwest in this rather nice spring day. And I'm hoping that you're having a good one as well. And this is me saying bye for now.